Thank you, Lisa. Thank you to the organizing committee. It's an honor to be here. And I've been asked to talk today about marine life for new medicines, human well-being and health. And as we've heard, oceans can provide food, energy, and all kinds of useful products, including medicines, as Timothy just said. And they can also produce a number of innovative products, such as proteins and enzymes, biopolymers, biomaterials, and I have two examples here. For example, marine glue, which is derived from the byssal threads of mollusks, and collagen, high-grade collagen, which is derived from medusae. But they can also produce new drugs, new medicines, and there are a number. Is there a light coming out of here? They can also produce very interesting new medicines. There's been a lot of success stories in recent years, and most of these, as you can see, have been coming from larger organisms such as sponges, cone snails, tunicates, and I'll be talking briefly about that in the future. But right now, what I'd like to talk to you about, I'm sorry if I have to switch back and forth, but I have a hard time seeing my screen in front of me. So I have to check my computer. But what's interesting is the number of drugs in general that come out on the market each year. The FDA approves a number of drugs, and here you can see the drugs that have been approved since 1993 to 2021. Where the pink part represents the new molecular entities, and the blue are biologicals replicating natural substances such as enzymes, antibodies, or hormones in our body. And you can see, like that there are ups and downs, but recent years, there's been an increase in the number of drugs that are marketed. And this is quite amazing that over the last 5 years, the annual average is around 51 drugs per year. And what is also amazing is that about 50% of the drugs approved are natural products. And by natural products are also called secondary metabolites. And these compounds differ from primary metabolites, such as proteins, amino acids, and fatty acids, which are the basic building blocks of life. But secondary metabolites are other metabolites that have survival functions for the organism. They help search for prey, not to be preyed upon, to locate mates, and so on, all kinds of communication functions. Often we don't know what the exact function of these compounds is and what benefits they confer to the producing organism, but we do know that they show strong biological activity in tests, in biological tests, and that's why about 50% of the drugs approved are natural products, and about another 50% are synthetic drugs that are often derived from natural products that have been engineered so that they are more functional, they are more aggressive to treating disease. And if we look at the drug discovery timeline of these drugs, we see that 450 million years ago, plants and animals developed chemical survival strategies, these secondary metabolites I talked about. And then a few million years later, humans discover medicinal properties of natural products by chance, by observing other animals, by bioassays. And then about 5,000 years ago, traditional medicine first recorded uses of natural products on Egyptian papyrus scrolls. Then we jump to the 1980s with the origin of synthetic organic chemistry. Active principles are identified and first attempts to synthesize important compounds like aspirin. Then we jump to the 50s, discovery of insulin, penicillin, streptomycin, and some other important antibiotics. And then after the 70s, we have DNA methods and molecular methods that are helping with high throughput screening, target drug design, and basically we're going towards the systems biology and what is known as personalized medicine. hopefully very soon. And whereas if we look at the drug discovery pipeline of marine-derived drugs, we've seen that this has been much slower. And that the first approved marine drug was approved in 1969 for the treatment of cancer, cytarabine. And then it took about another 20 years for the second compound to come out in 1991, and then about another 15 years for the third compound to come out. And then after 2005, more or less, we see that compounds come out at a much faster pace, about 4 or 5 years, if not less, until between one and the other. And this slide also shows that most of these compounds are to treat cancer, and this is quite interesting. And it also shows that the three compounds on the top part of the slide are presently being tested to treat COVID, because as Timothy was saying before, for now, there's only one FDA-approved antiviral drug to treat COVID, remdesivir. And so there's a great need to look for new drugs, and they're also exploring the possibility of new drugs from the sea. So currently, we have about 8 FDA-approved compounds, medicines from the oceans, with about 5 compounds that are in advanced clinical trials, and with about 50 compounds that are in clinical or preclinical trials. So these compounds should be coming out on the market quite soon. Why then is marine secondary metabolites important for drug discovery? Okay, we said that these secondary metabolites are defense attack signaling molecules, and this is true for terrestrial habitats as well. But what's interesting about the marine environment is that the chemistry of these compounds is totally different. And often these compounds have no terrestrial counterpart. So you have this high biodiversity in the ocean, which also translates into high chemical diversity. And according to the US National Cancer Institute, there's almost a 2 order of magnitude greater possibility of finding anti-tumor compounds from the oceans rather than from terrestrial samples. And this is why the compound annual growth rate of marine compounds is expected to grow very quickly in the coming years. So why does it take these compounds so long to come out in the market? Well, if you look at the left part of the picture, in the marine habitat, maybe we're testing 10,000 compounds in our bioassays. In terrestrial habitats, they're testing hundreds of thousand molecules at the same time. So let's look at our marine 10,000 compounds. From those, only about 250 enter into preclinical animal trial, okay? And from those, maybe only 5 compounds enter into clinical trial. And in the end, maybe only 1 compound will be approved by the FDA. And this has taken 15 years, which is much better than 20 years from a few years ago. So it takes a long time for drugs to be approved. And this is true for both marine and terrestrial, of course. But there are a number of bottlenecks in the marine environment that have also been a concern. And we tried to address this problem in the pharmacy project, EU pharmacy project, which ended a few years ago. And there were several issues. For example, there were problems such as physical access. Lisa, you talked about the deep ocean. It's easier now to maybe access the deep ocean, but it wasn't so in the past. And so this was a huge problem, limiting new chemistry, new individuals, and everything else. There are legal aspects which have already been talked about. And then you need novel biology, novel chemistry, and novel activity. You need novel chemistry because you don't want to come out with the same molecule that's already been discovered and which has gone through the pipeline and has been revealed not to work. So you need to know from the very beginning if you're dealing with something new or not. Unfortunately, we have programs for that now. And you need novel activity because you don't want to come out with a compound that already is very similar to compound that is on the market. And then you have problems of scale up and transfer, which are related to the fact that until now, drug discovery, as I showed you very briefly before, has been based basically on larger organisms such as sponges, tunicates, and whatnot, because these were easier to collect by scuba divers. They are difficult to grow in aquaculture. In fact, PharmaMar, which came out with trabactidine in 2007, had spent 10 years trying to grow this tunicate in aquaculture, but the animal was not producing enough of the molecule in order to go into clinical trials, and they only solved the problem when they were able to synthesize the compound. So now the big thing is to try to synthesize your compound before entering into clinical trials. It's also difficult to obtain sustainable biomass production without destructive collection. If you find a beautiful snail at 4,000 meters in the Central Ocean, what are you going to do to get enough biomass? You can't destroy the whole population. So then there are intellectual property concerns, which have been discussed before. So the trend now is to look at smaller organisms and the bioactivity, these smaller organisms such as fungi, bacteria, cyanobacteria, and algae. And for example, phytoplankton such as cyanobacteria, dinoflagellates, and diatoms are potentially the most novel source of bioactive secondary metabolites. They display all kinds of bioactivity and can be employed in the drug development and treatment. But unfortunately, for now, they're only at the preclinical and clinical trial stage. But they have the advantages that they're a single cell that can be grown to obtain large biomass in bioreactors, thereby avoiding destructive collection processes of larger marine animals. And this is the pipeline that, in fact, we use at the Stazione Zoologica. So we isolate our phytoplankton, upscale it in bioreactors, collect the biomass, do the chemical screening, do the cell screening to see what kind of bioactivity we have. And doing this, we've discovered several molecules. I'm just going to talk to you about two. One of these is amphidinyl. This is the new molecule from a dinoflagellate. Beautiful molecule, but too big, too expensive, too difficult to synthesize. And this is why we decided to publish this work in a specialized journal. Whereas in another case, we were able to, this is another compound derived from a marine diatom that has been engineered so that it's not like the original compound, very potent compound. It's been patented. And the patent belongs to Biosearch, which is a spin-off company of the Stazione Zoologica and the National Research Council in Italy. So in this case, it's being tested as an adjuvant for vaccines. For those who don't know what an adjuvant is a compound that boosts the activity of the vaccines. And it's also being tested as a booster for some chemotherapeutic drugs, and it's being funded by a regional program in the Naples area to go through preclinical trial. So now I'd like to move to another subject, very important, other health products from the sea for food and nutraceutical applications, which were referred to just before. Nutraceuticals are defined as any substance that is food or part of food that provides medical and/or health benefits, including prevention and treatment of disease. And here again, we see that there's a huge market for these compounds, and that most of the market consumption of these compounds is in Asia, in North America, but also in Europe. And I'm not going to talk about all of the nutraceuticals available because there are really thousands of them, but seaweeds is an example, which have long been known, especially by populations living by the sea that have integrated them into their foods for many centuries and knew that they had anti-inflammatory activity, for example, but they also have all kinds of other activities. So the trend now is to try to incorporate the seaweeds into functional foods. Not so much the single compound, but the whole organism, because sometimes it's the synergy between molecules that is more beneficial rather than the single molecule alone. And you can see they're being integrated in all kinds of beverages and whatnot, food products and whatnot. And the same is happening with phytoplankton. You have these green powders, which you can add to your favorite smoothies, yogurt, sugar, cereals, pasta, rice, oil, ice cream, and even give it to your dog because it's very rich in a lot of very good health-promoting components. Last but not least, let's plunge into the world of, sorry, I'm blocked here. Okay, into the world of marine cosmeceuticals. Here again, we see that there's a huge market for these compounds. Nutraceuticals are compounds that also have a health-promoting benefit. Oops, sorry. They're a cosmetic that has medicinal properties because, as anti-aging properties, because it doesn't just stay on the dermal layer, but it goes into the epidermis, okay? I'm sorry, from the epidermis into the dermal layer of your skin, and it transfers vitamins, enzymes, proteins, and all kinds of beneficial compounds into your skin. That's why they're called cosmeceuticals. Again, a very huge market for these compounds. So let me end by saying, I hope that I've shown you that the ocean is a sea of opportunities for human health products from the marine environment. Marine drug discovery is a fast-growing discipline, which includes not only the discovery of new ocean medicines, but also the discovery of new compounds to help prevent diseases, such as nutraceuticals and cosmeceuticals for aging, at least of the skin. The aim is to obtain a sustainable and continuous source of active compounds, example from microalgae that are more amenable to culturing in enclosed bioreactors compared to many macroorganisms such as sponges, corals, and mollusks, to meet the challenging demands of pharmaceutical industries. This would enable a more friendly, environmentally friendly approach to drug discovery and overcome some of the bottlenecks in drug discovery, such as the overutilization of marine resources and the use of destructive collection practices. The thrust in the end, both of clinicians and researchers, must now turn decisively towards prevention and cure, and not just cure, and oceans can help meet this challenge with new health products to improve human well-being in the future. And I thank you for your attention. Sorry about this miscoordination, but my eyes are not what they used to be until a few years ago. And I'll.
Thank you. We have a question over there in the corner, Marcelo. Oh.
Phosphorus come from fish, from some kind of fish. Phosphorus, and this is only for the fish. It's true, this.
Okay. Sorry, I don't know where the phosphorus supply is coming from humans. I would imagine it's coming from microalgae because they're easy to consume. You don't need to, obtain it from fish. You can obtain it from microalgae. There's been a huge revolution in creating in microalgae the biomasses you can obtain from microalgae, because for a while they were trying to build these huge bioreactors for energy, okay? as an alternative to fossil fuels, they thought they would be able to get the oils from microalgae to circumvent this problem, but it's economically non-viable yet. Okay, so with this type of research, they had all this technology which became available for phytoplankton growth, and this is why it's been channeled towards the production of nutraceuticals and other kinds of health benefit products, because there's so much that can be produced from phytoplankton of various types. And the phytoplankton, even we've been using the phytoplankton from the Gulf of Naples, nothing very exotic. So you don't even have to go very far. You're welcome.
Rashid has a question.
Yeah, so thank you. Are you people in this field, are you concerned about overfishing and what we do to the ocean? Is there a relationship between that and our potential here?
Yes, that's exactly the problem. I mean, like we're worried about the overfishing of these potentially, like very important medicines that from cone snails and sponges, and the tendency in the past to have, wiped out populations, whereas nowadays you don't need to do that. You can just isolate cells and grow them in the lab to huge amounts and do the testing from very little. It's exactly that, is to overcome destructive collection practices that were happening in the 60s and 70s when the first medicines came out.
Yes, Ariana, thank you for the wonderful overview. I have a question that addresses kind of what you say, but maybe takes it a step further, or I'm not so sure. I mean, the compounds are really interesting, and it's been around that research for quite some time, and some have been very successful, a lot of them not, but that's no reason to not do more of it. And you mentioned the importance of undiscovered parts of the ocean, really, in that frame. But on the other hand, there is increasingly regulatory and legal challenges around that. I mean, you haven't, I think, or maybe I didn't catch it, maybe you want to say a word about the Nagoya Protocol, about access to genetic resources, and the very or the answer that you gave on the last question is exactly one of the sticking points in the BBNJ agreement, that is intellectual property protection rights, and who's benefiting from, let's say, if a commercial entity makes a huge profit out of a particular organism, a strain or an element, and is the state of origin, let's say, the coastal arena, for example, where that sample was taken, entitled to benefit from that sharing. And I'm hearing, at least in my own community, in my own lab, a lot of discussion that legal frame around that makes it increasingly complicated to actually work outside of your own EEZ, which for many people is actually quite interesting. Thank you.
Okay, thank you. Very interesting problem that even for terrestrial plants and organisms has been a huge problem. And I think the pharma industry was very worried, and that's why they were very concerned about ocean medicines at the beginning. And it was only in recent years when they started seeing that we could get products from, like phytoplankton that is fished locally or collected locally, that you can get biologically active molecules just the same. Then we've demonstrated that you don't have to go to the high seas in order to, and there's still, like there still are going to be legal problems, because let's say I discover a compound from a species called Skeletonema that lives along the coast of Naples, but also along the coast of Turkey and, like most of the Mediterranean. And someone's going to say, well, why is your species different from mine? Why, so I'm sure there is going to be discussion about using smaller organisms as well. But this is, and honestly, I have not followed all of the legal issues regarding protection of property rights for, for these things. These are collections that we have in the laboratory for many years and that we are simply testing.